Okay, hi everybody, this is Lilith and I'm one of your IAs for BIBC 102. In this video, I will do a quick review over Lecture 16 and Lecture 17. Lecture 16 is containing most of nucleotide anabolism and a little bit of catabolism, and then Lecture 17 taps into a little bit of exercise physiology and how exactly metabolism is involved in exercise physiology. So, to understand nucleotide anabolism, let's first understand what nucleotides are and what nucleosides are. So, nucleotides are molecules that contain at least one phosphate group over here. They contain a sugar, which is a 5-carbon sugar, that's why it is called a pentose, right here. And it also contains a base that contains a lot of nitrogens. That's why they are often called nitrogenous bases. Now, this whole structure in blue is a nucleotide. A nucleoside is everything but the phosphate groups. So it only has a sugar and a base. Let's look at the different types of bases that can be attached to our sugar, right over here. There are two types of bases, purines and pyrimidines. An easy way of remembering which ones are the purines, see that the word purines is shorter and they match with the longer structures, and the word pyrimidines is longer, but it matches with the shorter structures, right? The one ring. Oh, the one rings instead of the two rings. Okay? The two purines that we have is adenine and guanine, and the three pyrimidines are cytosine, thymine, and uracil. Nucleotides are used in DNA, RNA. They are used for energy storage, like ATP, adenosine triphosphate, is a nucleotide. They are also used to activate other molecules. For example, if you remember back in glycogen synthesis, we use a molecule of GTP, guanosine triphosphate, to activate a molecule of glucose. Oh, I said that wrong. It's not GTP, it's UTP, I think. Check that. I believe it is UTP. To activate a molecule of glucose and attach it to the growing chain of glycogen. Okay, so that is where nucleotides are found. Now, there are two ways of making nucleotides that we discussed in class. First is the de novo pathway, which means basically starting from scratch, building from scratch. And that is when the base is literally built one atom at a time, and most of these atoms come from different sources. For example, these three come from glycine. This one comes from a random carbon dioxide. This one comes from aspartate. From my understanding, from what Professor Hampton said, you don't necessarily need to know exactly how that happens, except for the pathways that he briefly goes over. Okay, so even though I haven't included any pathways here, because most of it you don't really need to know to much great detail, you should watch this video in parallel with looking at the slides and the pathways that Professor Hampton included, just so you can follow the structures as I'm speaking about them. But there are only a few enzymes and reactions that you really need to memorize for this chapter. So the de novo pathway is when we build nucleotides from scratch, and the second way of making nucleotides is the salvaging pathway. And salvaging, as we already know what the word means, right? It's to get something else and to modify it into turning it into something new. It's exactly what happens. We use other existing molecules, other nucleotides, and we modify them into the one that we need. So let's begin to talk about the de novo pathway of making purines and pyrimidines. After we make these bases the de novo way, they later go on to become nucleotides. Let's talk about the purine first. So purine de novo synthesis uses the molecule PRPP, and it begins to build the purine base right on PRPP. In class, Professor Hampton mentioned that purine has the U, so purine is being made upon PRPP. Okay? And after we build the base of purine on the molecule of PRPP, after some reactions, we form IMP, which is inosinate monophosphate. And this is the first purine nucleotide. After that, it gets modified into the two other purine nucleotides, adenosine tri-di or monophosphate and guanosine tri-di or monophosphate. Okay, pyrimidine synthesis does not happen the same way as purine does. It does not build right upon PRPP. It builds over yonder, right? That's the example that Professor Hampton said. So yonder means over there. And by this, he basically meant that the base pyrimidine is built away from the molecule of PRPP. So then, when it is made, I'm just drawing a structure of what a pyrimidine could look like. After it is made, then it is placed onto the molecule of PRPP. Okay, so those are the two things that you want to distinguish between these two pathways. Purine synthesis is made right on PRPP, and pyrimidine synthesis, it first makes the pyrimidine away from PRPP, then when it is fully synthesized, then it is attached to PRPP. After these reactions take place, we make OMP, which is orotidine monophosphate, and this is the first pyrimidine nucleotide. Later, this gets modified into the three possible pyrimidine nucleotides. Okay, so that was the de novo pathway. Now let's look at the salvage pathway. The salvage pathway of making nucleotides for purines would use already existing purines, and we would add a molecule of PRPP, a few reactions, and we finally make the new nucleotide that we needed. To make pyrimidines, we use already existing pyrimidines, and instead of PRPP, we add a ribose 1-phosphate to make the new nucleotides. Again, do take a look at the pathways that Professor Hampton posted in lecture. I didn't put them here because it didn't make sense for me to go into that detail, but you should look at them, distinguish the differences between these two. If you do have any questions that are more specific to the reactions that are happening and you would like more detail, then I'm always here to answer with email or during my office hours or study session. Before we move on to purine catabolism, let's talk about the chemotherapeutic drugs that are specific to these purine synthesis or pyrimidine synthesis. So as we all know, chemotherapeutics are drugs that are against cancer growth, right? And what is cancer? Cancer is uncontrolled cell division. So the idea is if we can stop cell division, then we can stop the cancer from growing. So most of these drugs, or the drugs that we did discuss, are able to stop either purine synthesis or thymine synthesis, which thymine is a type of pyrimidine. This makes sense because if we stop purine synthesis and thymine synthesis, then we can no longer build viable DNA, right? We can no longer synthesize viable DNA. And if there's no viable DNA, then there will be no cell division, meaning the cancer growth should stop. So there are two types that we discussed in class. I'm sure there's many more, but these are the two that we talked about. So there are drugs that target this enzyme, glutamine aminotransferase. Okay, glutamine aminotransferase is an enzyme that takes the molecule of glutamine, which is one of our amino acids. Because glutamine has that amide group at the very end, the aminotransferase cuts off that amide and donates that nitrogen from the amide to the purine synthesis pathway, right? Because to make the purines, we need a source of nitrogen, and that source of nitrogen comes from glutamine. Now, if there is a drug that stops this enzyme from working, then this enzyme will never be able to give that nitrogen from glutamine to purine synthesis, and we will never make purine. Hence, we will not be able to make viable DNA. Okay? The other type of drug that we talked about inhibits the enzyme dihydrofolate reductase. Dihydrofolate reductase is an enzyme that is involved in the synthesis of thymine production. Thymine is a type of pyrimidine that is involved in synthesizing DNA, right? We need that to put it in the DNA. So if this enzyme inhibits dihydrofolate reductase, then the cell will not be able to make thymine, so it will not have good DNA, so cell division won't occur. Okay, so this is how these chemotherapeutics attack uncontrolled dividing cells. But it doesn't have to be exactly the uncontrolled dividing cells. As we all know, chemotherapy is also attacking healthy cells of the human body, right? So just a little insight into how chemo can actually affect the healthy tissue of somebody who has cancer. So, long story short, no nitrogen for purines or to build thymine. This means that we can't really build good DNA, meaning the cancer cells cannot divide. Okay. Purine catabolism. I guess the main takeaway would be that purines are broken down into this molecule called uric acid by the enzyme xanthine oxidase. However, this could be a problem for some people because when uric acid accumulates, mostly in our joints, it can cause a lot of inflammation and many other problems, and the person can develop gout. And at the end of lecture 16, you will see some pretty bad pictures of gout. However, there is medication that blocks xanthine oxidase. And hopefully this makes sense. It's a simple it's a simple inhibitory drug, right? If we stop the enzyme that produces the molecule that's causing problems, then that molecule will no longer be produced, and hopefully we will no longer have gout. Okay, and also, except these, diet also helps. Diet changes. Okay, I feel like I kind of breezed through this, so please let me know if you have any specific questions on any of the lecture slides, and I'll be happy to go over them. But do pay attention on specific spots that Professor Hampton told you to memorize, and others that he said, you really don't need to know this, because that could save you a lot of time. Okay. Lecture 17, we tap a little into exercise physiology. So we briefly talked about muscles, and I love this graphic. This is bone, tendon, big muscle. This shows exactly how there are many bundles, right, of these fascicles and many different muscle fibers, and even in this big thing, there are myofibrils, which are these specific proteins right here that slide along each other. So let's look at those. So we have the thick filament, which is myosin, and we have the thin filament, which is actin, and these two are proteins. The myosin has an important part on it. It has the myosin head, and this myosin head basically has an ATPase activity. ATPase, hopefully by now the name of the enzyme makes sense, right? Breakdown, ACE means breakdown, ATP. So the head of myosin is able to break down ATP, hydrolyze ATP, take the energy from it, and be able to grab on to the actin molecule and push it a little to the side. Now imagine if millions and millions of these proteins do this small push to the side, then the whole muscle or parts of the muscle can contract. Okay, so that's how contraction happens, and it all happens through ATP hydrolysis, because that is where the energy for these two to slide past each other takes place, or that's where the energy comes from. Okay. Then in lecture we talked about the two sources of ATP that we can use, one of them being glucose and the other being fats. So glucose is stored as glycogen, right? Mostly in our liver and our muscles. Glycogen is broken down by the enzyme glycogen phosphorylase, and it is broken down into single molecules of glucose 1-phosphate. However, this glucose 1-phosphate cannot leave the cell because it is still phosphorylated. So we either need to get rid of this phosphate for glucose alone to be able to leave the cell to go into the bloodstream and other cells, or it stays inside of whichever cell it is in. So if this glycogen store is in the muscle cells, the muscle cell does not have the enzyme glucose 6-phosphatase. So what will happen is glucose 1-phosphate will turn into glucose 6-phosphate through phosphoglucomutase, and then the muscle will just break glucose 6-phosphate down through glycolysis. Okay, it will not be able to give free glucose to any other cell or the blood. Okay, so long story short, if the muscle has the glycogen, it will break it down into glucose 1-phosphate, then into glucose 6-phosphate, then it will break it down further through glycolysis and get the ATP from it. However, if these glycogen stores are in the liver is the giver, right? So liver actually will be able to cleave this phosphate group off of the glucose and make uncharged glucose, right? Glucose without the phosphate, through the use of, this is phosphatase, I didn't mean to erase that. Basically, the liver has the enzyme glucose 6-phosphatase, which is able to take off this phosphate off of the glucose. Okay, and now glucose is uncharged, and it is able to be released into the blood and go to other cells. That is why liver is the giver. Okay, fat stores are mostly in our adipose tissue. And one important thing about fats is that fats contain a higher capacity of energy because they produce more molecules of ATP per molecule of fat versus glucose, right? If we think about it, if we take a molecule of glucose and we break it down through glycolysis, we will make 2 molecules of ATP, right? And even if it goes through a Krebs cycle and oxidative phosphorylation, I believe we will make 38 ATP, or 35 to 38, something like that. But if we take a molecule of fat, which on average could be around, I don't know, 9 to 14 carbons, or however many carbons, and we just produce way more ATP by breaking down 1 molecule of fat. So it has more capacity than glucose. One topic that we covered that is kind of apart from ATP and glucose and fat storage is RQ. RQ is respiratory quotient. And RQ tells us what type of energy storage an individual is using at a point during their existence, right? So the quotient talks about the amount of carbon dioxide produced over the amount of oxygen consumed. So if this person's RQ is 1, this means that they are producing 1 molecule of carbon dioxide for every 1 molecule of oxygen they consume. If this is the case, then they are using only glucose as their source of energy. When would this be the case? This would be the case if I go outside to get food and I see a lion and the lion's gonna eat me and I have to start running, right? I will sprint for my life, right? At that moment, I will be using only glucose because my glucose in my bloodstream would go through glycolysis in my cells and it will give me rapid ATP, right? At that moment, my RQ will be around 1 because I will be using solely glucose and possibly also creatine phosphate, but we'll talk about creatine phosphate later, okay? However, if somebody's RQ is around 0.7, this means that they are producing way less carbon dioxide molecules for the amount of oxygen they consume. If this is the case, then they are using only fats. When would this happen? Well, this is most likely happening to me right now. As I sit at my desk, I'm relaxed, there's no lion around me, I'm talking to my iPad, making this video. And why is this happening? Because I am able to actually breathe, right? So I'm consuming a lot of oxygen. And I'm not using a lot of energy, right? I'm not aggressively breaking down glucose because I'm running away from a lion. I'm just sitting here. All that's moving is my hands, right? So I'm not producing as much carbon dioxide as I would be if I was running away from a lion. So right now my RQ could possibly be around 0.7. And this is if somebody's using only fats. These two are the measurements, the extreme measurements, right? Let's say if I go out and I start going for a walk, a slow walk, my RQ could be around 0.85 maybe, right? Somewhere in the middle. This will mean that I'm using a little bit of glucose and a little bit of fat to get my ATP. Okay, so hopefully those concepts made sense. And basically RQ tells us what type of ATP or energy source is an individual using in a given time. The other concept that we talked about is VO2 max. VO2 max is the maximum rate of oxygen consumption. For example, this is how much oxygen can I consume per amount of time, right? And VO2 max is different for every individual. A person who is more trained has a much higher VO2 max because they are able to take up way more oxygen in, let's say, 1 minute than I am, right? Because I'm definitely not trained right now. So the trained athlete versus me, the trained athlete will be more used to breathing deeper, breathing faster, right? Because they're more trained. So their VO2 max will be much, much higher than mine. Two types of VO2 maxes that we discussed in class was the absolute VO2 max and the relative VO2 max. Absolute VO2 max is calculated by the milliliters of oxygen consumed over however many minutes that you are being measured consuming these oxygens. Okay? Relative VO2 max is the same equation for absolute VO2 max. However, we also divide the milliliters by kilograms of the individual. This helps to compare two different people who are at different weights, right? Who have different masses. For example, if I want to be compared, so this is me, right? I'm only 5'6", and if I want to be compared to a person, let's say, who is 6'4", right? Let's say, right? Giant person, I wish I was 6'4". Then they will have a much bigger mass, right? Maybe they have a bigger lung capacity. Maybe they have bigger lungs. I don't know. But if we only calculate our VO2 maxes with absolute VO2 max, then this individual will clearly have a much higher VO2 max than I will, because they simply can take up more oxygen. So this ratio for them will be much larger. However, to make it a fair comparison in science, when comparing two individuals, we will use relative VO2 max, because this way we can divide this ratio by the kilograms of the individuals, and this way we will equilibrate the VO2 maxes, and we can compare them more fairly. Okay, let me delete my artwork before I'm too embarrassed. Okay, so hopefully those two types of VO2 maxes made sense and how they are used. The next concept we talked about is steady state oxygen consumption. So steady state happens when we are able to consume oxygen at a constant rate, right? For example, if I go outside and I start walking at a pretty fast pace, at first, I'm going to feel like I'm out of breath, right? Because I'm just starting to pick up walking. However, if I keep walking for a minute or two, I will realize that my breathing is becoming more controlled. I don't feel like I'm going to pass out, right? I've gotten used to it. That most likely means that I have hit steady state of walking and oxygen consumption. Okay? It means I'm breathing constantly, and I'm probably burning the same amount of energy more constantly. Okay? So this graph shows that exact what I just explained, right? I'm sitting in my chair, and then I get up and I say, I'm going to start walking. But when I start walking very fast at first, I start to have an oxygen deficit, right? Because although my muscles are moving, I'm walking very fast, I can't keep up with the oxygen demand. So I feel like I'm running out of oxygen until I walk for a while and I finally feel like I'm not going to pass out. I finally hit steady state of oxygen consumption. Okay? During the time when I am having oxygen deficit, I am using a lot of anaerobic activities, a lot of anaerobic respiration. So maybe most of my muscles do use creatine phosphate or do glycolysis. And then when I start to come around to the steady state oxygen consumption, then I am able to have enough oxygen to do oxidative respiration. For example, Krebs, oxidative phosphorylation, and also fatty acid breakdown, because all of those happen in the mitochondrion, right? Okay. Yeah, so here I wrote out another example. If I start to run right away, I will be using the glucose storage first. And I should probably add that I will also be using the phosphocreatine storage also. And if you remember from lecture, phosphocreatine is basically the molecule creatine that is phosphorylated, and it is just sitting in my body when I'm not doing any quick movements. But when I do have quick movements, then I use up all of the ATP that is readily available, right? So I use up the ATP. Now this is ADP. And to be able to replenish this, to be able to use this ADP and turn it back to ATP, I have creatine phosphate that basically gives its phosphate to ADP to form ATP rapidly. So you can think of creatine phosphates as like the emergency phosphate donor. When I do some quick movements, my levels of ATP deplete and I need to replenish them. That is when creatine phosphate comes in and says, hey ADP, take my phosphate, replenish the ATP. Okay? Here I have drawn out the pathway of glucose. So the glycolysis portion of glucose breakdown is oxygen independent. We don't need oxygen to be able to do glycolysis. However, we need oxygen to be able to do Krebs, ETC, and oxidative phosphorylation. This means that the breakdown of glucose through glycolysis is much faster, right? I can get up and start running, and I will be doing a lot of glycolysis, and I'll be breaking this glucose down, and I'll make a lot of pyruvate. But because in that first 2 minutes of me running very fast, I'm not getting enough oxygen just yet, I will not have enough oxygen to take this pyruvate and completely break it down through Krebs and OXPHOS. And because of that, the pyruvate molecules will be shunted over to lactate dehydrogenase enzyme to form a bunch of lactate molecules. Okay, this is why if you do, let's say, what is it called, a HIIT program or a HIIT workout, right? Or if you go to the gym and you lift weights, then the next day your levels of lactate in your blood will be elevated, because the HIIT and also lifting weights is a very anaerobic exercise, right? So you'll be doing a lot of glycolysis and probably almost none of that. So you'll have a lot of lactate in the blood. Now that brings us to our next topic of lactate threshold. So lactate threshold is the precise amount of lactate that your muscle cells can have before the lactate starts to basically come out of the muscle cells and spill over into the blood. Okay? And the more trained you are, the higher your lactate threshold will be. Now, what determines this exact threshold? So when we produce a lot of lactate through this pathway, when we're doing a lot of anaerobic exercise and using glycolysis, these lactate molecules have to be processed by biochemical reactions, or else if they all stay in our cells, then, this will be like wasting carbons, right? There's 3 carbons over here, right? So these are processed by reactions. For example, some of it is turned back into pyruvate. And then, pyruvate can be used to do gluconeogenesis, right? Lactate can be turned into pyruvate, and then pyruvate can be turned into amino acids, right? The cell can do a lot of things with lactate, but the main point is it needs to be processed. However, if your muscle cells aren't used to this exercise, then you're not going to have many of those enzymes that perform the lactate processing, right? And if you don't have those enzymes already built in your muscle cells, then a lot of the lactate that is produced isn't going to be processed. So it will basically ooze out of muscle cells. So let's look at this graph and see that as you are doing an exercise that increases your usage of oxygen, right? So, for example, if I am just walking slowly, then that will be at 25% of my VO2 max. But if I decide to start running away from a lion, that will probably be at the higher ups of my VO2 max, right? Because I will be consuming a lot more oxygen. I will be exercising at a much higher strength. However, there is a point in your VO2 max where your muscle cells will no longer be able to process lactate. And once you hit that threshold, lactate levels in your blood will start to rise more rapidly. So as you see here, this is the untrained person. Their lactate threshold comes at a much lower VO2 max, and the trained person's lactate threshold comes at a much higher VO2 max. Again, because they have trained so much, they have done this exercise so much, that their muscle cells are more ready to process lactate before it spills over into their blood. Okay, hopefully that topic made sense. And the last thing that I want to talk about are the 2 types of, is this the last thing? Yes. The 2 types of muscle cells that we can have. Well, technically 3, but type 1 and type 2. So type 1 muscle cells are what are called the slow twitch muscle cells. And these are named slow twitch because these types of muscle cells are more prominent in people who are able to run slower, but for longer periods of time, right? And type 2 muscle cells are the fast twitch muscle cells, which are more developed in individuals who can do more quick and more strong exercises. Okay, so let's look at some of the points for type 1. So type 1 muscle cells have low ATPase activity of myosin. If you remember what I said in the beginning of chapter 17, ATPase activity is what the myosin protein has in the muscle cells, right? So if this type of myosin uses less ATPase activity, then it will be using less ATP to basically do weaker muscle contractions, I want to say. I'm not sure how accurate that is, but hopefully I'm painting the right image in your head, that because these are slow twitch muscles, they will be having less ATPase activity. So they won't need to contract muscles as fast. Okay? They will have slow calcium handling. And calcium is, we didn't really stress this enough in lecture, I thought, but calcium is used in muscle contraction. So again, if we don't really want any fast type of muscle contraction, then low calcium or slow calcium handling is appropriate for type 1 muscles. Less well-developed glycolytic capacity than fast switch fibers. And this makes sense also, right? We said that glycolytic pathway is the faster pathway. It uses, it doesn't need oxygen. It is oxygen independent. So it makes sense how the muscle fibers that are slow twitch don't really need that many that many glycolytic pathways. And also, what's very prominent about type 1 fibers is that they have larger and more mitochondria. And this makes sense because, as you already know, the pathways that take place in the mitochondria are oxygen dependent, right? They need oxygen to take place. And so type 1 muscle cells would be found in, let's say, marathon runners. And as we all know, marathon runners run for hours, right? And they need a lot of oxygen. So it would make sense that their muscle cells have a lot more mitochondria to be able to do all of those oxygen-dependent reactions. Okay. I won't go over type 2. It's basically the opposite of type 1. And I got these from your lecture slides, so you should just look over them and understand the differences, right? You don't necessarily need to memorize these, I don't think. Just understand the differences. This graph is good because it shows the maximal power of each type of muscle fiber. And true to all of these four, you can see that type 1 muscle fibers have very low power compared to type 2s. And this makes sense because we don't want marathon runners to be necessarily very strong and deadlift like 300 pounds, right? We need them to run longer, be more enduring to the pain or to the lengthy run, right? But people who have type 2 muscle cells, these are the fast twitch, right? These are the people who are the 100-meter sprinters in the Olympics, right? These are the people who do deadlift like hundreds of pounds because they need to do it quick and fast, right? So they have more type 2 muscles, which have a lot more power. I'm not sure if it was mentioned in class, but type 2A and type 2X, they're just two varieties of type 2. I think most normal people who aren't like Olympic either sprinters or endurance runners, most people have type 2A, and then these are more of the extremes. Extremes in good way, not bad way. Okay? And one last topic I want to cover is the difference between power and capacity. So hopefully this power versus capacity makes sense by now, but let's go over it. Power pertains to the pathways and the reactions that give us quick ATP, right? And power is needed if I need to lift something that's a couple 100 pounds, right? Because I need to do it fast and just be done. I need a big surge of power. and the power is going to come from either phosphocreatine or glycolysis. But as we move down this way and we get to Krebs, OXPHOS, or beta oxidation, which is breakdown of fats, I get more high capacity, right? So it's not as powerful, right? If I use only fat breakdown and I metabolize glucose only through Krebs and OXPHOS, I will most definitely not be able to lift a couple 100 pounds, right? I'll be able to run for 2 hours because that has a lot of capacity, but I won't have enough power. Okay, so the more to the right you go this way, the higher capacity you get. And the more you go this way, the higher power you get. Okay, hopefully these topics made sense. Please don't rely too much on this video and do look at the lecture notes. I think lecture 16 and 17 are quite different than other lectures that we've gone over because these aren't technically, like, we're not learning whole biochemical pathways, right? This is more application and understanding the concepts and just knowing a few steps here and there. So do look at the PowerPoint for those steps that you need to memorize, and then just try to understand the main concept of everything else. Okay, as always, if you have questions, especially during this time before finals, please email me. Don't hesitate. My name is Lilith. You can find this and the notes in my IATA folder. I will do an extra office hour / study session tomorrow, December 12th at 5 p.m. So if you're watching this before then and you have questions, do come by. And my office hours on Monday at 7 will still hold. And yeah, that's it for now. Watch out for another video on the last 2 lectures. Good luck studying, everybody.